the living organism are composed of, which are the main component of the living organism. That every living organism consists of one or more cells. A cell is the smallest unit of life, means from a single cell, the division of cells occurs, and many cells, they just evolve from one cell to multiple cells. Now, that is why cell is called the smallest unit of life, because the single multicellular organisms are there, as well as the unicellular. Uni means one. So the unicellular organisms, that is the bacteria, these are the unicellular organism. So the cell is the smallest unit of the life, individually alive when as part of a multi-celled organism. Either they can survive on the earth individually, or they can be existed in the earth as the part of a multi-celled organisms. Both ways cells can survive. Number 3 point is every living cell come into existence by division of a pre-existing cell. Means first one cell is there, and from a pre-existing cells, multiple cells, they generate by division, cell division. The somatic cell divides by mitosis, and the germ cell, that is the sperm and the ovum, they divide through meiosis. There are two types of cell division. Number 4 point is cells contain hereditary material, that is mainly the DNA, that they pass along to their offspring. Offspring means their children. Offspring means the next generation cells which have been evolved by division of the parent cell. So during the process of cell division, DNA as the hereditary material, they are passed from one generation to another generation. So these 4 points, they compose the cell theory. If question comes, what is the cell theory, and question comes mostly of 3 and 4 marks, then you have to write the cell theory of 1, 2, 3, 4 points. Now we will be just moving
Excuse
to
me, ma'am. Ma'am, the link is not opening. It is showing that we are sorry we can't open the link. Means you which PDF you sent me, it was showing that we can't open it.
Which one?
The mail ones you which we sent it. The
Mail
book.
means, oh, okay.
The book one.
Let me finish the class. Then only book will be solved. Let
Okay.
me finish.
Okay.
The
Okay.
class link is okay, na? The class link is okay, na? So
Yes,
let me.
class means the book you mailed me, Albert.
Oh, Baba. Please wait.
Okay, okay,
When
ma'am.
I finish the class, then I will be expediting that link. Okay.
Okay, okay, ma'am.
Maybe it is shared in the Google Drive, na? So
Yes, ma'am.
maybe
It's
the
in Google Meet.
authorized, so it is a large book. So authorization maybe I have to give. So I will solve later. Please, if there is no question regarding the PPT or the matter, please wait. At the end, I will be
Okay, okay,
solving
ma'am.
everything.
Okay, ma'am. Sorry for
So
that.
now the prokaryotic cells. What are the prokaryotic cells? The presence or absence of nucleus. This is the nucleus, the main genetic material, that is the here it is the DNA. The organelle of the cell, it is the single cellular organism, means only one cell is there, not multiple celled organism it is. These bacteria, you can see this is the picture of a bacteria. It is the single celled organism where the cell wall, the yellow color is the quite thick cell wall, and underneath the cell wall, this gray color outer line is the cell membrane, and these gray color and entangled portions of the cell organelle, it is the genetic material, that is called the DNA. What is the full form of DNA? Deoxyribonucleic acid. And the dots here, it is the ribosome. Ribosome is another component of cell organelle which help to transcribe the codon, means what is the genetic material available. Let me go backward. Here you can see the DNA synthesis replication. The slide is changed. The slide is changed.
Yes, ma'am.
Okay.
Yes,
So
ma'am.
when you study the DNA replication, why it is needed? The DNA component, the genetic material component, at the time of cell division, they have to also be divided. They have to also be multiplied. If you not increase their volume, when the cell will divide, then the genetic material will be halved. And then when they will again divide, the genetic material in comparison to the parental matter, it will be, the quantity will be the 1/4. So it won't suffice the need of the passage of the genetic material from the parents to the offspring. That is why the replication of the genetic material, whatever it is there, it should be replicated. The DNA should be, the content of the DNA should be increased first, then only the division will be done. Otherwise, it will be halved. The conception is clear? So, at the time of DNA replication, in the next step, from DNA to RNA, this synthesis matter, it is called the transcription. The ribosome is the cell organelle which help in transcription. And after transcription, from RNA to protein, the building block of the DNA, they are comprised with different amino acids. And these amino acids, the decoding of the amino acids happens like this, from DNA to RNA and from RNA to protein. The more complex, the macromolecule, the ATGC, these are the smaller component, they are the micro one, and when they form a protein molecule, it is called the macromolecule. So the translation happens in the last step, and the whole process from DNA to RNA, actually it is mRNA, and from RNA to protein, it is called the central dogma. And in the transcription process, the ribosome, it is the main cell organelle which helps to transcribe the strands of RNA. Now, if you just look into a unicellular bacteria, then you can see there are the most important organelles are only there, not very difficult or different type of variety types of organelle which are existing in the eukaryotes. They are not available in the prokaryote. Only the main one are there. And another thing is the flagellum. They help them in their locomotion. So the main characteristic feature of prokaryotic cell is few microns long, no nucleus, no intracellular organelle. Now, if you go to the eukaryote, I will be showing the previous slide later. The eukaryotic cells, they have different type of organelles. So you just look, eukaryotes, the word comes from the Greek word eu, means well or truly, and karyon means the nucleus. So a well organized nucleus is the characteristic feature of the eukaryotic cell. And they have the other unique features which differs from the prokaryotic cell. It is the length, because their size is larger in comparison with the pro one, that is the tens of microns long. They have profound, well organized nucleus, the diverse type of intracellular organelles, That is, there are compartmentalization. There are different compartments within the cells, and single-celled or multi-celled organisms like plant, animal, or human, they have eukaryotic cells. Though the amoeba, yeast, they are single celled, but also their cells have some well-organized nucleus. And the multi-celled organisms like plant, animal, and humans, they obviously contain the eukaryotic cells. Now, what are the organelles you can see? Let me zoom this one. Now, this is the nucleus. This nucleus have the in between dark, dense, composed, roundish appearance. It is called the nucleolus. The nucleus have DNA called chromatin. When the DNA is entangled, they look like bundles of wool. As if you just unwind the wool, how it looked like? The DNA in a crude nature inside the cell just look like this way. There are entangled wool or threads like this. For transcription and translations, this entanglement has to be unwinded, and the double-stranded DNA, they are made single-stranded, and from the single-stranded DNA, in the stepwise, the RNA is transcribed. Now, the nucleus, if you just look vividly, this nucleus is encapsulated with double layered membrane. It is called the nuclear envelope. There are two layers. This green color, can you see the cursor? Can you see the cursor?
Yes, ma'am.
Okay.
Yes, ma'am.
So this double layered nuclear envelope, they have some gaps in between. Gaps. Just see, there are nuclear pore. This nuclear pore helps to go inside different cellular matters inside the nucleus or go outside the nuclear matter when required. There are other cellular organelles like mitochondria. These longer stretched, and inside there are compartments like this. We will be showing vividly the internal compartments of the mitochondria. So these are mitochondria. These are filaments, actin filaments. These actin filaments, they give the skeletal structure. As we have bones, over the bones, the muscles, the skin, they have been put to give you a structure, defined structure. So the cells, the eukaryotic cells also possess some actin filaments which give them some specific structure. There are other organelles like Golgi apparatus. These brown colors, there are vesicles and multivesicular apparatus. It is called the Golgi apparatus. There are plasma membrane. There is, not are, because only one plasma membrane which just covers the eukaryotic cell. It is the plasma membrane. And there are mitochondria, I told, lysosome. This yellow color vesicle, it is the lysosome which carries different lytic enzymes. When something is needed to just digest, these lytic enzymes, they are released from lysosomes. They have lytic capacity. Lytic capacity means cleaving capacity. So they can digest anything through the lytic enzymes inside them. There are vesicles, different type of vesicles, vacuoles, and microtubules, centromere. There are centromere. You can see there are sky blue color, two centrosomes inside this one, and there are actin-like filamentous structure of these centrosome. These centrosome, they help in cell division. At the time of cell division, the centrosome with their centromere, the genetic material or the DNA, they are attached with them and they help them to divide the genetic matter into two subcells in perfect manner. So mainly these are the eukaryotic cellular organelles which are not present. All of them are not present into the unicellular prokaryotic cell. They are more simple. Now I want to show you a video. If you see the video, then it will be very clear to you. Then I can just go to the next slides. Are you interested in videos, or I should just continue in the slides?
We are interested in videos.
Okay, let me share the video. What are the difference between the prokaryote cell and the eukaryote cell? This is the video. Why it is not coming in the full screen?
Prokaryote.
Are you seeing?
Yes, ma'am.
Sound is coming. Sound is coming.
Yes, ma'am. Sound is clear.
Yes, ma'am.
No, video sound.
Yes, it was
হ্যাঁ
coming.
ম্যাডাম ভিডিও সাউন্ড শোনা যাচ্ছে
আচ্ছা আমি একটু আস্তে করে দেবো কি একটু বেশি জোর মনে হচ্ছে লাগছে নাকি let me start.
This distinction between a eukaryote. When we want to categorize life as we know it at a very high level, we can categorize it as either eukaryotic, or as a eukaryote, or as a prokaryote, prokaryote. And the largest distinction between a eukaryote and a prokaryote are membrane-bound structures that eukaryotes have that prokaryotes don't have. And the most noticeable of which is a membrane-bound nucleus. So in a eukaryote, the genetic information is going to be inside a membrane-bound nucleus. So this right over here, this is the nucleus. This is the nucleus, and you have your genetic information inside it. You have your DNA. Here, for prokaryotic cells, you're going to have your DNA, and it might be bundled up into a section of the cell. We call that a nucleoid. But it's not membrane-bound. So let me write this down. No membrane. Let me write it. Membrane-bound nucleus. But that's not just the only distinction between eukaryotes and prokaryotes, although it's the one that's most noted. In fact, the word eukaryote, the karyote part, comes from the Greek for nut or kernel. So let me make this clear. So this is referring to a nut, or a kernel, while prokaryote means before the nut or kernel. So we don't see, and I guess you could think of the nut or the kernel as the membrane-bound structure, especially the membrane-bound nucleus over here. But that's not all. Eukaryotes will also have other membrane-bound structures that you will not see in prokaryotes. For example, you will tend to see mitochondria in a eukaryotic cell, both plant and animal cells, but you won't see it in a prokaryotic cell. And there's other types of membrane-bound structures. You could see things like Golgi apparatus. And this over here is a micrograph of a eukaryotic cell. And you see the Golgi apparatus right over here, which helps package proteins. You see a micrograph of mitochondria. This is a micrograph of the nucleus. So this right over here is the nuclear membrane. And then you see all the genetic information. It's all spread out. It's in chromatin form right over here, but you see it's especially densely packed right over here. And we've also depicted it in this right over there. So this is all the DNA in chromatin form, but this part that looks extra dense or dark in this micrograph right over there, we call that the nucleolus. We call that the nucleolus. And we now know that this is where ribosomal RNA is being produced. And ribosomal RNA, that forms part of the structure of ribosomes, which are essential in the translation, or I guess you could say the construction of proteins based on the information in mRNA. And we can go into a lot more depth in that in other videos. So these are ribosomes right over here, and they're made up of ribosomal RNA, and they're also made up of proteins. And so this nucleolus over there, that's where that's happening, that denser part of the nucleus. So the key distinction, eukaryotic cell, you have a membrane-bound nucleus. You have other membrane-bound structures like mitochondria. In fact, there's some theories that mitochondria first evolved as prokaryotic organisms that eventually lived in symbiosis inside of a larger eukaryotic cell. And then the other distinction is that in eukaryotes, the DNA tends to be in multiple strands. So the DNA, if you were to kind of straighten it out, it would be in multiple strands, while in a prokaryote, the DNA tends to be circular. It can be all flipped around and whatever else, but at the end of the day, it would be circular DNA. So those are the three core distinctions: nuclear membrane, other membrane-bound organelles like mitochondria and Golgi and the Golgi apparatus, and then you also have multiple single strands of DNA versus circular DNA. Other things is that eukaryotes tend to be larger, while prokaryotes tend to be smaller, and they tend to be simpler. So now that we know the key distinctions, what are examples of eukaryotes? Well, eukaryotes include most of what we interact with on a daily basis, or we think we're interacting with on a daily basis. This includes all multicellular organisms. So multicellular organisms. So I'm thinking animals, plants, fungi. It includes protists. This is a paramecium right over here. This is eukaryotic. It's going to have a membrane-bound nucleus and other organelles. This right over here, these are onion root tip cells. So these are plant cells, and you can actually see it's been stained. You can actually see the membrane-bound nucleus here. And this is actually a cool picture because you can see these cells at different stages of mitosis, which is interesting. Animal cells, the things that make you are eukaryotic. So what is prokaryotic? Well, bacteria is probably the most common example of that. Bacteria, right over here. These are prokaryotes. And lesser talk, or the thing that's talked about a lot less is Archaea. And Archaea, people initially thought that these were a form of bacteria that just lived in very extreme conditions, but now they know that it's a completely different domain of life. Archaea. And so when we think about the domains of life, the current thought is that you have bacteria here, you have Archaea, and then you have eukaryotes. I'll do it there. And then you have eukaryotes. And these are things that have all of the traits that we've talked about. And so these include plants and animals and fungi and unicellular eukaryotes, protists and things like that. And so if we, once again, just high level, we would consider these prokaryotes, and these, of course, are the eukaryotes. So hopefully that gives you a good overview of things.
Wait, I am sharing another one. I'm not finding that one in the tab. Video on Prokaryote.
So, as you know, all cells can actually be split into two categories. You've got your prokaryotic cells and the eukaryotic cells. And the difference between the two is that the eukaryotic cells have a nucleus and the prokaryotic cells don't have a nucleus. Now, we've already talked all about eukaryotic cells and their structure, and you can rewatch those videos again by clicking here. Bing! But in this video, we're going to be talking about prokaryotic cells. Now, animals, plants, fungi and prototists are all made up of eukaryotic cells, but we believe that actually they all evolved these more complex organisms from these simpler cells that lack a nucleus, which are called prokaryotic cells. So really these are where life started as far as we're concerned. As you will see from this presentation, prokaryotic cells have some features in common with eukaryotic cells, but they also have a lot of differences. All bacteria are examples of prokaryotic cells. So really what we're really essentially the majority of prokaryotic cells are which are extremely hot. They're called extremophiles, these bacteria that live in these, really very important in biology, and they're sometimes underrated, but they're actually really interesting. They can be found in a huge range of environments, places like deep sea vents, which are extremely hot. They're called extremophiles, these bacteria that live in these conditions. So that's very interesting. They also keep our ecosystems going because they're responsible for decay of organic material. A lot of them are pathogens, which means that they cause diseases, not only in humans, but in lots of other living organisms as well. We can also exploit them in biotechnology. We use bacteria for things like yogurt production. A bacteria called lactobacillus is used to make yogurt. They contribute significantly to our health. We've got lots of them living inside our guts, our microbiome, which help with digestion and keep our guts healthy. Did you know that actually 1 kilogram of your body weight is actually made from bacteria, and of your body more cells are foreign bacterial cells than they are actually your own. You can actually exchange them very easily between each other, which is why people get ill if we exchange pathogenic bacteria, and actually it's pretty prokaryotic cells will be covered in the next 2 videos. This video is going to be looking at the structure of prokaryotic cells, and the next video is going to be on a little bit on antibiotics, looking at penicillin, how penicillin works, reproduction in prokaryotic cells. The 2 main types are ones called cocci, which means round, or bacilli, which means rod-shaped. Now, this word usually forms part of their binomial name. For example, things like lactobacillus are rod-shaped ones, or staphylococcus are round-shaped ones. Now, the first thing to mention on prokaryotic cells is that they're actually very small. Their diameter is between 1 and 5 micrometers. Now, if you compare that to a typical eukaryotic cell, they can be up to 40 micrometers, anywhere between 20 and 40 micrometers. So prokaryotic cells are a lot smaller. You can see that in this lovely picture here, which is of a squamous epithelial cell, a human cell. You can see the pink nucleus in the middle there, and then the cytoplasm surrounding it. And all these little worm-like purple shapes here are actually lactobacillus bacteria. So you can see just how much smaller they are than a typical eukaryotic cell. Prokaryotic cells all
Are you getting what is the video going on?
Yes, ma'am.
possess a cell wall, a cell membrane, cytoplasm, DNA and ribosomes. And some prokaryotic cells also contain a capsule. Some of them have infoldings of their membranes. Some have plasmids, flagella and pili. So we'll cover all these structures in this presentation. So starting with the cell wall. Now the function of the cell wall is to prevent the cell from bursting, and it gives bacteria its shape. It's made of a substance called murine, which is a peptidoglycan. Now peptidoglycan is a combination of polysaccharides and proteins. There are actually two types of cell wall, and we'll be discussing them later on in this presentation. The cell membrane is pretty much exactly the same as you'd find in a eukaryotic cell, and again we've done a video on that one which you can watch by clicking the link here. Boom. The cytoplasm is also very, very similar to eukaryotic cells. I suppose the major difference really here is that there is no cytoskeleton. Remember, prokaryotic cells don't have a nucleus. They do still have DNA. It's very important that they still have DNA. It's one single chromosome. It's not in a membrane like the nucleus. It doesn't have an envelope around it. That's why there's no nucleus.
So what is told here, that the prokaryote have genetic material as nucleoid. So the entangled DNA-like genetic material, they are carrying the hereditary matter. But as they are not a well-formed nucleus, they are not called nucleus. They are called the nucleoid. And when the nucleus composed of nucleolus, they are encapsulated with nuclear envelope, and the well-formed organization is done, then it is called the nucleus. But the prokaryotes, though they have genetic matters, but don't have the well-composed nucleus.
Here it's just free in the cytoplasm, this nucleoid floating around, and it's not associated with any proteins. Normal eukaryotic DNA is associated with histone proteins. This DNA is not. It's what we call naked DNA. Prokaryotic cells also have ribosomes, like eukaryotic cells, but there is a difference here. They're actually smaller than the ribosomes found in eukaryotic cells. They are what we call 70S ribosomes rather than the 80S ones found in eukaryotic cells, and these ones are made up from a 30S and a 50S subunit. Their function is the same as in eukaryotic cells, which is, that's right, protein synthesis. Now some prokaryotic cells also have this extra layer around the outside called a capsule or a slime layer. It's made from various different molecules such as starch or glycolipid, and it protects the bacteria from drying out, which is called desiccation, which is very useful in certain environments for certain bacteria. It can also help the bacteria from being discovered by the immune system, and it prevents infection by various viruses like the bacteriophage, which we're going to be looking at a bit later on in this presentation. It can also help them adhere to certain surfaces. So a capsule can be very, very helpful to the survival of certain prokaryotic cells. Some prokaryotic cells, membranes actually fold inwards, and that sort of increases the surface area, which is probably for some kind of metabolic reaction. It was thought it was for respiration instead of mitochondria, because obviously prokaryotic cells don't have any membrane-bound organelles. But now it's maybe thought it might be more likely for photosynthesis in photosynthetic bacteria or nitrogen fixation. Now some prokaryotic cells also have small circular loops of DNA called plasmids. Now plasmids are very, very interesting. They can actually be transferred between bacteria in a process called conjugation, and they code for a specific trait. Some bacteria have flagella for movement, almost like a little tail that comes out the back of the bacteria. And it's made from some protein fibers called flagellin, which are spun around like a motor. It's a spiral protein fiber, and it gets spun around by a little motor protein, which helps with the movement of bacteria. Other bacteria also have these little things called pili around the outside, and these are used to help bacteria reproduce in a particular way, and also communicate in a process called conjugation, where they will be able to link together, join together, and exchange these small little plasmids. So a lot of exam questions will actually get you to compare the structure of prokaryotic cells with eukaryotic cells. And a good way to do that is with a table. So why don't you pause the video here and copy and complete this table. Now, as already mentioned in the video, there are actually two types of prokaryotic cell wall. This was discovered by a Dutch scientist called Christian Gram in 1884, and he developed this specialized staining procedure which showed up that there were these two types of bacteria. So one of which he called gram-positive bacteria, and one which are called gram-negative bacteria. Now, gram-positive bacteria have a very, very thick layer of peptidoglycan, which contains lots of stuff called teichoic acid. Now, this traps a particular part of the stain called crystal violet, which means that these end up being a lovely purple colour. The gram-negative bacteria have a much thinner layer of peptidoglycan, and they also have this thing called the outer membrane. And that means that actually the crystal violet stain gets washed away very, very easily. Now, they will pick up a stain after they've been washed in the staining process called saffronin, and that gives them a more kind of red pinkish colour. So you should know the basic sort of steps for this staining procedure. Some examples of gram-positive bacteria, things like lactobacillus or micrococcus luteus, and examples of gram-negative bacteria are salmonella and E. coli.
Let us join our PPT. You can see my PPT.
Yes, ma'am.
So is it easier to have a video than explain through PPT?
Yes, ma'am.
I
It is
think,
easier.
yeah, it will be easier. So what you have just noticed in the prokaryotes, they are different from the eukaryotes, and the just leave now the prokaryotes and eukaryotes. Whatever you have known that there are some differences regarding the well-organized cell organelles in the eukaryotes and not in the prokaryote. Now we are coming to the functions of the cell organelles, that is the cell membrane. Cell membrane is the outer layer of the cells. The animal cells only have cell membrane, but the plant cells, they have both the cell wall, and beneath the cell wall there are cell membrane. There is cell membrane. Now the functions of the cell membrane is, number one is receiving information, that is outside the cell. If there are different changes happening, they just interact with the receptor over the cell membranes, and the information is passed to the molecular level cell signaling pathways inside the cell. So receiving information. Now import and export of molecules. So there are need of export import of molecules, ions in between the different cells, and this is done by the cell membrane. Number three function is capacity for movement and expansion. So for going here and there, suppose it is a unicellular, uni means single cellular. So the capacity for movement, unicellular organelles, they have to move. Their locomotion is functioned by the only cell. So the movement and expansion to just expand in their size, to divide, all these functions, they are done by plasma membrane or the cell membrane. Now the organelles.
Excuse me, ma'am. Ma'am, sorry
yeah.
to stop you, but ma'am, we have a electric laboratory class from 4. So the class has already started in the MS team. So can you please leave?
Okay, how can it start? Because it is not 4 yet.
But
Ma'am, yes,
yes,
it is 4.
it is 4, ma'am.
Okay, let me stop. Okay, bye.
Ma'am, take
Thank you,
attendance.
I have requested to take the attendance. Has it been recorded? The organizer can only
Yes, ma'am.
record
It has
that.
been recorded.
Okay, okay.
Thank you.
Okay.
Ma'am, about the permission in the mail.
I couldn't hear you.
Ma'am,
Stop
about
recording.
the
Stop recording. Now stop recording. We can have a discussion. Whenever